Your questions for mutations in DNA repair? Okay. The first question that I want to go through is for how can a mutation in a region of DNA that does not code for a protein affect gene expression? So you should be able to give me 2 examples. No, 3, but yeah. Sorry, go ahead. Well, we did talk about mutations where it's in the splicing region where you don't splice it correctly, and then that will affect gene expression. So that's one of them. Yeah. That would be, but that would be a mutation within that gene. So, but yeah. But you're right that mutations in introns that lead to incorrect splicing would affect gene expression. So that's one of them. What would be another non-coding region that you can mutate that would affect gene expression? So we talked about 2 more. Right, so mutation in the promoter, that could result in making too much protein or not making enough. And then what's the other one that we discussed? What's that? So more general than that, in any type of regulatory DNA site. Okay, so if you have a regulatory piece of DNA, meaning when I say regulatory piece of DNA, I'm talking about regulating the expression of a gene. Okay, so there's certain segments of DNA that control the expression of genes, and if you have mutations in those regions, those can also affect gene expression. Can someone talk about the significance of a mutation in germ cells? Remember, germ cells are also called germline cells. What's that? Right, they can be passed on to offspring, correct. And then number 8, mutation in a gene in the fruit fly Drosophila was found to affect eye color. The protein affected was found to be completely normal in its molecular weight and amino acid sequence. Although much less of the protein is made in mutant flies, the most likely reason for the change in levels of protein is that the mutation occurred where? So you have to use all the information to kind of figure this one out. No, and the reason that it's not D is because if it affected the splicing of the mRNA, that would change the molecular weight and the amino acid sequence of the protein, and they told you both those things were normal. So the fact that the molecular weight and amino acid sequence is normal rules out a lot of them. It's not C. So if you had a frame shift mutation near the start of the coding region, frame shift mutation, it would still hold on. There's a frame shift mutation near the start of the coding region. Okay, so sorry, this that's from the book. So what they're saying is let's say you have an insertion or deletion that occurs before the start codon, that would actually not affect, it wouldn't lead to producing less protein. Right, A. So, and the reason that it could be A is that let's say the mutation in the promoter decreases the likelihood of RNA polymerase binding to the promoter. That would lead to making a decreased amount of mRNA, and then that would lead to making a decreased amount of protein that mRNA codes for. Okay. Number 10, which of the following would occur from a mutation in the gene's promoter region? Right, E. So the rate of transcription may be increased or decreased because of that. Number 11, a researcher determined that a strain of E. coli is producing a shortened version of a protein required for glucose metabolism. What type of mutation could be responsible for this shorter than normal protein? Right, A, nonsense. So remember, a nonsense mutation is going to change a normal codon into a stop codon, and if you do that, that means you stop making the protein sooner than you should, and that's going to lead to a shorter version of that protein being produced. Okay, so number 12, what type of gene mutation occurred to produce the following protein sequence? So I added this part in here. The protein sequences written below are written using the one-letter abbreviations for amino acids. So each letter represents one amino acid within this sequence of amino acids. So what type of mutation occurred in this case? What's that? Right, in this sense, because it only changed one amino acid. And then 13, so I give you the normal gene and then the mutated gene, and then both the normal and mutated protein. So based on that, what type of mutation occurred? No. It was an insertion, but what did it cause then? What type of mutation did the insertion cause, looking at the protein sequence? Right, right. So it's an insertion, and that insertion resulted in a frameshift mutation. So basically what happens is every amino acid after the site of insertion is incorrect. So you have methionine, alanine, glycine in both of them, and then here was, in the nucleotide sequence, there's an insertion here, and then every amino acid after that is incorrect. 14, somatic cell mutations are heritable. That's false. Right. If it read germ cell mutations are heritable, that would be true. Do you guys see other questions within this topic that you guys want to go through? Okay, so we're going to move on to a new topic then, which is gene regulation. And what we're talking about in gene regulation is actually discussing how gene expression is regulated. Okay, so basically we, previous to talking about mutations in DNA repair, we talked about how gene expression, how that process takes place. And then now what we're talking about is how a cell regulates that process to either end up producing the protein that the gene codes for or not producing the protein that the gene codes for. So when we go through this topic, I'll say the gene is on or the gene is off. And just to remind you that if the gene's on, that means that the cell is making the protein that that gene codes for. And if the gene is off, then that means the gene is not making the protein that that gene codes for. And then in terms of expressed versus not expressed, a gene that's on is being expressed, and a gene that's off is not expressed. Okay, so a cell is not expressing every gene within its genome. It's only expressing a certain subset. And so we're going to kind of learn some basic rules about how a cell determines whether it's going to express a gene or not. We're going to spend most of our time talking about simple examples found in bacteria because they're easy to understand and they provide you with a foundation for gene regulation. And then we'll talk about some differences in terms of how genes are regulated in eukaryotic cells. There's similarities, but then there's also some important differences. So that's what we're talking about in this topic. We're talking about regulating gene expression. And this is a picture taken from your textbook. And each red arrow is supposed to represent a point at which you can regulate gene expression. Okay, so you can regulate gene expression in a bacterial cell at the level of transcription, at the level of translation, and then you can also have post-translational control of gene expression. And then this is showing you a eukaryotic cell and showing you all the points at which you could affect gene expression. So those are all the points at which you could affect whether the protein is going to be produced and be functional or not. Okay? And in terms of this picture, the width of the arrow is how prominent that form of regulation is. So you can see that in both cell types, the arrow pointing to regulation of level transcription is the widest. That means you get a lot of regulation at that level. So basically what you're deciding at that level is either are you going to transcribe or are you not going to transcribe. Obviously if you don't transcribe, then you're never going to produce the protein. If you transcribe it, then you have the possibility of producing the protein. And then there's variances on that. There's also how much are you transcribing the gene, so the rate of transcription is also affected at this point of regulation. So is there, are you completely inhibiting it or just decreasing the amount? And then are you increasing it, and how much are you increasing it? Okay, so at the level of transcription, what you're deciding in this class in terms of just an easy way to think about it is, are you going to transcribe it or not? Okay, and that's what we're going to spend most of our time talking about, is how regulation of gene expression occurs at the level of transcription. And the reason for that is because that's the most prominent form. Okay, so we're going to talk about gene regulation in bacteria first. And before gene regulation, talking about gene regulation in bacteria, or to do that, we're going to talk about two operons. One is called the lac operon, and the other is called the TRP operon. Okay, so the first thing we're going to do is define what an operon is. And operons are found in bacterial cells. Operons are not found in eukaryotic cells. Okay. Okay, so we're going to talk about how the lac operon and the TRP operon are regulated. We're going to talk about it in a lot of detail, and this will provide a great foundation for understanding how genes are regulated, some basic rules for gene regulation. Okay, so just looking at this, this is how we kind of described when we discussed transcription. We say that you have a promoter, then you have your, let's say this is the gene, and then you have a terminator. So you basically have a promoter, RNA polymerase binds here, transcribes the gene, and then on binds when it hits the terminator. And so that's how we kind of discussed it. But in bacteria, you can have the genes organized into an operon. Okay, and so what you have in an operon is you have a promoter, and then instead of only having one gene after the promoter, you have multiple genes. So in this example, you have genes E through A, and so and then there would be a terminator at this end. And so what happens, and that's what an operon is. And what happens is RNA polymerase would bind, transcribe all these genes together, and then produce this mRNA molecule that carries the coding sequences for every gene that is under the control of that promoter. Okay, so this is the definition of an operon. So it's a cluster of contiguous genes, and that just means that the genes are right next to each other. So E is right next to D, right next to C, the B to A. So it's a cluster of genes that are transcribed from one promoter, and it's going to give rise to a polycistronic mRNA. We described polycistronic mRNA last week, but just to remind you, a polycistronic mRNA is an mRNA molecule that carries the coding sequences for multiple genes. or you could say that it carries the coding sequences for multiple proteins. Okay, so this would be how you produce a polycistronic mRNA. You produce a polycistronic mRNA by transcribing an operon. Okay, and one of the advantages of having operons, so one of the advantages of having one promoter controlling the expression of several genes, rather than having them each controlled by their individual promoters, is that the different genes that are a part of an operon are involved in the same process. Okay, so we will discuss it for the TRP operon and then also the LAC operon. But if they're all involved in coding for proteins that are involved in the same process, all you have to do is upregulate this one or express this one operon rather than having to individually express every gene that may be involved in producing a protein that's involved in that pathway. So you kind of upregulate them all together by expressing this one operon rather than having to express the genes individually. Okay, so that's kind of what an operon is. In terms of what it consists of, an operon is going to consist of a promoter, and these are all segments of DNA. So you have a promoter, you already know what that is and what it does. So you have a promoter, you're going to have an operator, which is also a segment of DNA, and this is an example of a regulatory segment of DNA. And so the operator is going to regulate the expression of the genes that are a part of the operon. Okay, so the operator's role is that it's a segment of DNA that controls the expression of the genes that are a part of the operon. And we'll see how it does that for both the operons that we talk about. And then you have a set of genes, and that's going to change depending on which operon you're talking about, so in terms of the number of genes. But then you're going to have a set of genes, one right after another. And then obviously at the end of that you would have a terminator, and you already know what that does too. So an operon is going to consist of these different segments of DNA. And then we'll see that when we talk about the LAC operon, there'll be additional segment of DNA that we'll talk about. But these are the ones that both operons have in common: a promoter, an operator, a set of genes, and a terminator. Okay, so also when we discuss the regulation of operons, and this will also be important for regulation of genes in general, there's two types of proteins that can be involved, and their names pretty much tell you what they do. One type of protein is called a repressor, and the other type of protein is called an activator. And they both are going to exert their effect by binding to DNA. So repressors and activators are proteins that bind to DNA. A repressor is a protein that binds to DNA and inhibits transcription or decreases the rate of transcription. And an activator is a protein that binds to DNA and increases the rate of transcription or allows transcription to take place. Okay, so we're going to see how these are involved in regulating the operons that we discussed, but activators and repressors also exist in eukaryotic cells and are involved in controlling gene expression. Okay, and again, it's key that in order to exert their effect, they have to be bound to the DNA. Okay, so we're going to see when we study these operons, we'll see two repressors, and one thing that's going to change is when is the repressor bound to the DNA inhibiting transcription and when is it not bound. And it has to be bound in order to inhibit transcription. Okay, so those are activators and repressors. So the first thing we're going to do is study the regulation of the tryptophan, the TRP operon. So this one, because it's easier, it's the simpler example. And TRP stands for tryptophan. And tryptophan is an amino acid. So the bacterial cell, and we were talking about this is an operon that's in a bacterial cell, any organism would need tryptophan because it's one of the amino acids you would need to make proteins. And so the genes that are a part of this operon are all involved in coding for proteins involved in synthesizing this amino acid. Okay, so we're going to see that, you can see it in this animation that we're going to watch here. You have genes E, D, C, B, and A. All those genes code for proteins that are involved in synthesizing tryptophan, this amino acid that the cell is going to need. Okay, and so basically what we're going to see is that a bacterial cell could find itself in two environments with respect to tryptophan. So it could find itself in an environment that has tryptophan present. So I'll write that as plus tryptophan. So plus tryptophan means that the bacterial cell is in an environment where tryptophan is present. Okay, and then minus tryptophan means that the bacterial cell is now in an environment that lacks tryptophan. Okay, so if you're a bacterial cell, just using what these genes code for, when do you think this operon is going to be expressed? And when do you think it's not going to be expressed? Right, so it's going to be on, meaning the operon will be expressed, and you're going to transcribe it when you lack tryptophan, and then it's going to be off when you have tryptophan present. And that makes sense because if you have tryptophan present in the environment, then the cell can just use that tryptophan. It doesn't need to make it. And it would be a waste of energy in terms of the bacteria to express these genes involved in making something you already have. So when you have tryptophan present, we're going to see that transcription doesn't take place. And then we're going to see that when the environment lacks tryptophan, then transcription is going to take place, because if you don't have it in the environment, then you do want to make it because you need it to build particular proteins within the bacterial cell. So this is a relatively simple regulation. We'll watch this animation describing it, and then we'll also talk about it on the other side of the video.
In the bacterium,
Let me turn that up a little bit.
In the bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is made, but it is unable to bind to the DNA and block transcription. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. Tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription.
Okay, so that's a relatively simple switch in terms of either you're going to transcribe these set of genes or not. So again, just to repeat some things that I've already said, so the TRP stands for tryptophan, and it's going to consist of the promoter. So that's this segment of DNA right here. You can see that the operator, the segment of DNA that's going to control gene expression of these genes, sits within the promoter. And then you have your set of genes, and then what's not shown in the picture is the terminator that would be at the end of gene A. And then again, we said that each of these genes code for a protein that's involved in producing tryptophan for the cell. Okay, so again, with respect to tryptophan in the environment, you only have 2 options. You either have tryptophan or you do not have tryptophan. So this is showing you what happens when you have low or no tryptophan. So they said what's regulating the expression of this gene is a repressor, and that's called the tryptophan repressor. Okay, and when there's no tryptophan present, the repressor cannot bind to the tryptophan repressor cannot bind to the operator, and then if the tryptophan repressor is not bound to the operator, that means RNA polymerase can bind and transcribe those genes. Okay, so the tryptophan repressor here in this case, as you can think of as off, because there's no tryptophan bound to it, and there's no tryptophan bound to it because the environment lacks it. And so now RNA polymerase can bind to the promoter and transcribe those genes. And so then, but if you find yourself in an environment that has tryptophan, then what's going to happen is the tryptophan is going to bind to the repressor, and you can think about it as activating it. And what that means is with tryptophan bound to the repressor now can bind to the operator. Okay, so the only way that the tryptophan repressor can bind to the operator is if tryptophan is bound to it. So then the repressor binds to the operator, and that prevents RNA polymerase from transcribing those genes. Okay, so this is a relatively simple switch, and you can see that the tryptophan is going to control whether the repressor is active or not. So when you don't have tryptophan, the repressor is inactive, and then when you have tryptophan, the repressor is active. And then tryptophan is going to control the repressor by binding to it. Do you have a question?
Yeah, are the repressor and the activator, are they enzymes?
No.
Okay.
They are just proteins that bind to DNA and will inhibit transcription. Okay, and then also one thing to note is that in terms of regulating this operon, there is no activator involved. There'll be an activator involved in the regulation of the LAC operon, but not the TRP operon. That's one reason that this is simpler because you only have a repressor involved, and with the LAC operon, you're going to have an activator and a repressor being involved. So essentially, you're going to have two switches for that operon. You shouldn't think, no. Because tryptophan is not a protein. Tryptophan does not bind to DNA. And what you would call tryptophan is a co-repressor. Because what happens is, so a co-repressor is a small molecule that binds to a repressor and activates it. And so tryptophan being a small molecule in an amino acid does that. So if you wanted to call it something, you would call tryptophan a co-repressor. No. If you want to. I think it's referenced in some of the questions that I have in lecture questions, so that's why I kind of brought it up. But yeah, you'll see it. You might see it in one of the lecture questions, but I don't require that you know what it is. Okay, so the next thing we're going to talk about is how the LAC operon is regulated. And LAC is short for lactose, which, if you remember, lactose is a disaccharide. And all the genes that are a part of this operon code for proteins that are involved in allowing a bacterial cell to use lactose as an energy source, meaning they're involved in the bacterial cell taking it up, breaking it down, and using it to produce ATP. Okay, so all the genes code for proteins involved in using lactose as an energy source. And then with this operon, a bacterial cell is only going to want to use lactose if it doesn't have glucose. Okay, so if it has glucose, it prefers glucose and will use that instead of lactose. So in terms of this operon, there's actually going to be 4 conditions that we're going to be considering. So we have one condition where you have glucose, that's plus glucose, and the environment lacks lactose, that's minus lactose. The second condition is that you have glucose, and then you have lactose in the environment, so you have both. A third situation is that you lack glucose, and then you also lack lactose in the environment, so you lack both. And then the fourth situation is that you lack glucose, but now you have lactose in the environment. So those are the 4 possible conditions with respect to these 2 nutrients in the environment. Okay, and then what we're going to see in terms of regulation of this operon is that it's off in these first three situations. And that makes sense because you're only going to want to express those genes if the cell has lactose, if it's present, but it doesn't have glucose. Okay, so it's only going to want to express these genes if it has lactose but no glucose. And so the only situation in which this is going to be on is in the minus glucose plus lactose situation. Okay. We're considering all these situations off, but to be more realistic, in some of the situations you do get a low rate of transcription, but we're just going to consider a low rate of transcription to be off, and then on, in this case, means a high rate of transcription. Okay, so what we're going to do is watch this animation, and you'll see that the regulation of this operon involves both an activator and a repressor. And basically what happens is the levels of glucose are going to regulate the activator, and the levels of lactose regulate the repressor. Okay, so the levels of glucose are going to regulate the activator, and the levels of lactose are going to regulate the repressor, and we'll see how the combination of the two will result in the expression of these genes.
The lac operon in Escherichia coli bacteria has a coding region for 3 proteins necessary for the use of lactose as a food source. These proteins are only needed when lactose is available and the normal food source, glucose, is in short supply. Therefore, the regulatory region of the lac operon has 2 switches. One, the cap binding site, is switched on only when glucose levels are low. Another, the operator site, is switched on only when lactose is present. Both of the switches must be on to allow expression of the lac operon genes. The cap binding site is next to the promoter for the lac operon. When the level of glucose is low, a high level of a compound called CAMP builds up in the cell. A complex of CAMP and a protein called CAP binds to the cap binding site, bending the DNA and allowing RNA polymerase to bind at the promoter. When the glucose level is high, there is not enough CAMP, so nothing binds at the cap binding site. This keeps RNA polymerase from binding at the promoter, thus preventing expression of the lac operon genes. The operator site is also next to the promoter. When lactose is absent, a repressor protein will bind at the operator site and prevent RNA polymerase from binding at the promoter. When lactose is present, a lactose isomer will bind to the repressor and prevent it from binding to the operator site. Thus, only when the glucose level is low and lactose is present will RNA polymerase bind to the lac promoter and carry out transcription.
Okay, so that kind of took you through a regulation of the lac operon. So again, you can see that there was an activator involved, and that is called CAP. I'm not going to ask you what CAP stands for, but it stands for catabolite activator protein. And this is the activator that's involved in regulating this operon. And then you had the lac repressor, and that's obviously the repressor that's involved in regulating this operon. So you have both the CAP and the lac repressor regulating whether this operon is going to be expressed or not. Okay. Also, when you look at this operon at the top of the picture here, there's one additional segment of DNA that you should be aware of, and that's the CAP site. Okay, and the CAP site is called that because that's the segment of DNA where this activator binds. Okay, so CAP binds to the CAP site. And so you can see that this operon has that CAP site. It also has a promoter, an operator, a set of genes, and then a terminator. Okay. Another thing you have to be aware of in order to kind of understand how this operon is regulated is this side reaction that happens in cells when lactose is present. And they're showing you that on the board here. So what this bacterial cell wants to happen is it wants to take lactose and break it down into galactose and glucose. So that's the main reaction. But there's this side reaction that happens inside the cell where you change lactose into this lactose isomer called allolactose. Okay, so in the animation, they referred to a lactose isomer, and that lactose isomer is called allolactose. Okay, and this is going to be involved in regulating the repressor. Okay, that's why it's important, and that's why we're bringing it up, because allolactose is going to regulate the repressor. Okay, so essentially, you need, in terms of the activator and the repressor, you need two things to be true in order for this operon to be expressed. Okay, so in order for this operon to be expressed, you're going to need cap bound to the cap site. Okay, so one thing that has to be true is that cap has to be bound to the cap site, and then also the repressor cannot be bound to the operator. Okay, so in order for this operon to be on or expressed, you need cap bound to the cap site, and you need the repressor not bound to the operator. Because anytime the repressor is bound to the operator, it's going to be off, and anytime that cap is not bound to the cap site, we'll see that's going to be off. Okay, so this is just taking you through, this is from your book, taking you through how the repressor acts. So this molecule here in the picture is the lac repressor. And so this is showing you what happens in terms of when there's no lactose. So what they're showing you here is that if there's no lactose, then the repressor is active and it'll bind to the operator. Okay, so any situation where you don't have lactose in the first and third situation here, the lac repressor is going to be bound to the operator, inhibiting transcription. Okay, so anytime that you lack lactose, you're not going to get transcription because the repressor is going to be bound to the operator. And then when you have lactose present, then I told you that there's this side reaction that produces allolactose, and I told you that allolactose is going to control the repressor. It does that by binding to it. Okay, so allolactose binds the repressor and inactivates it, which means that now the repressor cannot bind to the operator. Okay, so any situation where you have lactose present, then you're not going to have the repressor be bound to the operator. Okay, but that doesn't necessarily mean it's going to be on, because remember, you also need cap bound to the cap site. Okay, but any situation where lactose is present, the lac repressor is not going to be bound to the operator. Okay, so this then kind of includes how the operon is regulated using both cap and the repressor, and it has all four situations on here, so I'll kind of connect them to how I drew them over here. So this is the plus glucose plus lactose situation. So in terms of this one would be situation number 2. So the plus glucose plus lactose is the left-hand corner. And the end result is that the genes are off, the operon is off in a situation. And so what we're going to do is talk about why. So because we'll start with glucose being high. Actually, let me take a step back. Oh no, we'll talk about it now. Glucose levels are going to control CAP, and they do that by controlling the levels of cyclic AMP. Okay, so what happens in a bacterial cell is glucose inhibits the production of cyclic AMP. And if you remember back to the animation, cyclic AMP is a small nucleotide that binds the cap and activates it so that it can bind the DNA. So in order for cap to bind to the DNA, it needs cyclic AMP bound to it. Okay, so if you have glucose inhibiting the production of this molecule, that means that cyclic AMP is not going to be bound to cap, and cap is not going to be bound to the cap site. Okay, so in this situation where you have glucose being high, that means that it's inhibiting the production of this nucleotide, which means that cap is not going to have cyclic AMP bound to it, which means that cap sits over here and it's not bound to the cap site. Okay. So one reason that this is off in the situation is because the activator is not bound to the cap site. And then the lactose levels are high, or you have lactose, so that means that you're going to have allolactose present, and it's going to be bound to the repressor inactivating it so that it will not be bound to the operator. But again, this operon is going to be off because cap is not bound in this situation. Okay. And then this situation down here at the bottom is plus glucose minus lactose, so that would be the number one situation here, so plus glucose minus lactose. So again, similar to this situation up here, because you have glucose present, it's inhibiting cyclic AMP production, which means that it's not going to be bound to cap, which means that cap cannot bind to the cap site. Okay. And then in addition to that, because you don't have glucose, that means there's no allolactose around, which means that the repressor is active and bound to the operator. And so in this situation, it's off for two reasons. One, the repressor is bound to the operator, inhibiting transcription. And then in addition to that, cap is not bound to the cap site where it can activate transcription. Okay. And then we're going to go down to the lower right-hand corner here. This is a situation where the environment lacks both nutrients. So that would be the third situation here, minus glucose minus lactose. So in this situation, because you don't have glucose, then that means you are going to be producing cyclic AMP. Cyclic AMP will be bound to cap, and so cap is active and bound to the cap site. But then because lactose levels are absent or low, that means that you do not have allolactose around, which means the repressor is active and bound to the operator. So in this situation, you have the activator there and working, but the repressor is bound to the DNA and it prevents transcription. So it's off because the repressor is preventing transcription from taking place. And then the top right-hand corner is the only situation in which it's on, and that would be the minus glucose plus lactose situation. And so again, because you have no glucose, that means that you're going to have cyclic AMP bound to cap and cap bound to the cap site. And then because you have high lactose, or you have lactose, you're going to have allolactose bound to the lac repressor, inactivating it and preventing it from binding to the operator. And now you have a situation where cap is bound to the cap site and the repressor is not bound to the operator, and then you're going to get this operon transcribed at a high rate, or we'll just say that the operon is on in that situation. Okay, do you guys have any questions about what's happening in any of the scenarios? Okay. So that's the lac operon. Okay, so this kind of summarizes how activators and repressors can regulate transcription. So when you look at this picture, this is obviously not taken from your textbook. On the left, you have negative regulation, and that can obviously happen through repressors. And then positive regulation happens through activators. And then what it's showing you is different ways that repressors and activators could respond to a small molecule. Okay, so when you look at the top left-hand corner here, what they're trying to show you here in this picture is that the repressor is active and bound to the DNA when it has no small molecule bound to it, but then a small molecule binds to the repressor and removes it from the DNA so it can't bind. Okay, so one of the repressors that we studied works like that. Do you know which one? So one of the repressors that we talked about When it doesn't have anything bound to it, it binds to the operator, but then when a small molecule binds to it, it's prevented from binding to the operator. Nope. What? Yeah, it's obviously the lac repressor one. So what's the small molecule that binds to the lac repressor that prevents it from binding to the operator? Right, allolactose. Okay, so in that case, the red arrow here would be allolactose binding to the lac repressor. And then this is showing you how a different way that it could be, and this is how the tryptophan repressor is regulated. So the repressor is active when it has a small molecule bound to it, and it's inactive when it doesn't have the small molecule. So this is obviously the tryptophan repressor. What's the small molecule? Right, tryptophan. Okay, and this is showing you two ways that activators could be regulated by a small molecule. And we only talked about one activator. So what you have up here is you have an activator that's active when there's no small molecule bound to it, and then it's inactive when a small molecule binds to it. Yeah, someone keeps on turning on the lights on and off. And then down here, you have an activator that's active when the small molecule is bound to it, and non-active when it's missing the small molecule. So we only talked about one activator, which was CAP. Which one describes how CAP is regulated? Right, the bottom one. And what's the small molecule that binds to CAP to activate it? Right, cyclic AMP. Okay. So this just describes how activators and repressors can provide for positive and negative regulation. And it's also showing you how small molecules can affect whether the repressor or the activator is active or inactive. Okay? So one of the things that you can see in bacteria for both the operons, and this is true for gene expression in general for bacteria, is that you get a lot of changes in gene expression in response to changes in the nutritional environment that the bacterial cell is in. So what nutrients does it have and which nutrients does it lack are going to affect which genes are expressed in bacteria. Okay, and then when we study eukaryotic gene regulation, activators and repressors are also involved in that form, or sorry, in us and other eukaryotic organisms. Okay. Okay, so regulation of transcription in eukaryotes follows some of the same principles as those found in bacteria. However, some important differences also occur. So in terms of similarities, some of the similarities that you should know for this class is that you're also going to have segments of DNA that control the expression of genes. And those segments can control the expression of genes by binding either activators or repressors. Okay, so that's kind of stating two similarities, sorry. One similarity is that there are regulatory segments of DNA that control gene expression. And then another similarity is that you're also going to have activators and repressors, and those two are obviously connected because activators and repressors are going to regulate gene expression by binding to those regulatory segments of DNA. Okay. Some important differences that you need to know for this class is one, the regulatory pieces of DNA that affect gene expression in a bacterial cell are typically right next to the genes that they regulate. Okay, and that's not true necessarily for eukaryotic organisms. The regulatory segments of DNA that control gene expression can be really far away from the gene that they actually regulate. And we'll see that in some cases the regulatory segments can also be what you would call upstream of the gene. They could be downstream of the gene. There's also some regulatory segments that sit within introns of genes. Okay, so then one difference is that the regulatory segments of DNA that control gene expression can be far away from the genes that they actually control the expression of. Okay, another difference that occurs is if you remember back to talking about how transcription occurs in eukaryotic cells, we talked about the fact that it involves these things called general transcription factors. And we said that the general transcription factors have to assemble and they mediate the binding of RNA polymerase to the promoter. And so there's examples of regulating gene expression in eukaryotic cells where you regulate that assembly process. So you regulate the assembly of those general transcription factors, and through regulating that process, you can determine whether a gene is going to be expressed or not. Okay, and then the third important difference that you need to know for this class is the fact that you can also have regulation of gene expression by affecting how the DNA is packaged in a eukaryotic cell. Okay, so you can affect how the DNA is packaged in a eukaryotic cell, and that can affect whether the gene is going to be expressed or not. So remember, the DNA is wrapped around and complex with proteins, and you can affect how they're complexed with those proteins, and that can end up regulating gene expression. Okay. We'll skip this slide, so I'd love to skip this slide. And this slide also. We're not going to get into what's called the Tata box, or how this kind of refers to how this is involved in regulating the assembly of the general transcription factors. Okay, and then one way that activators and repressors may influence the regulation of gene expression is by influencing the function of GTFs, which is general transcription factors. So that's just one way that activators and repressors can function to affect gene expression, is by affecting the general transcription factors. Also, there is this protein called mediator that's involved in transcription in eukaryotic cells. We're not going to get into what mediator does, but activators and repressors may also influence the function of this mediator, which you need present and active in order for gene expression to occur. And then you can also get transcription being controlled by changes in chromatin structure. And essentially what you have in this case is you're going to have a change in chromatin structure that makes the DNA more accessible to proteins, okay? Meaning it's more accessible to proteins binding to the DNA. Okay, so if you make the DNA more accessible to proteins, that is something that could increase the rate of transcription. Conversely, we'll see that you could inhibit gene expression by making the DNA less accessible to proteins. So if you make the DNA less accessible, then the proteins that are involved in transcription can't access the DNA, and that would be a way of inhibiting transcription. Okay, so again, another way of controlling transcription in eukaryotic cells is through controlling chromatin structure. And again, when this happens in a positive way, what you're doing essentially is making the DNA more accessible to the proteins involved in transcription. And if you're going to inhibit it, it will result in changes that make the DNA less accessible to the proteins involved in transcription. And we're not going to get into this in detail, but one way of doing that is by modifying the histones. So if you remember, we talked about in eukaryotic cells, the DNA is wrapped around these histone proteins, and there's modifications that happen to histones that can either increase or decrease the rate of transcription, and they do that by affecting the chromatin structure. Okay. So this is what I just mentioned. So you can have histone modifications that affect gene expression. There's other ways other than affecting chromatin structure that histone modifications could affect gene transcription. But for this class, you just have to know that modifying histones can affect whether a gene is going to be expressed or not. Another thing that regulates gene expression in eukaryotic cells is DNA methylation. And so what you're doing in DNA methylation is you're actually just adding methyl groups, which is one of the functional groups that you needed to know at the beginning of the course. It's adding methyl groups directly to DNA. Okay, so DNA methylation, or if you methylate DNA, that's adding methyl groups directly to the DNA. And as it says in the slide there, DNA methylation is usually going to inhibit the transcription of eukaryotic genes. Not always, but usually when you methylate the DNA, that's going to result in inhibiting the transcription of the gene or genes. Okay. And then, do you guys have any questions so far about regulation? The last thing we're going to end with is talking about regulation at the level of translation. So everything that we've talked about now, we talked about regulation of transcription in bacteria, and then in terms of eukaryotic organisms, everything we've discussed so far is also regulating expression of genes at the level of transcription. And then we're going to talk about one way of regulating the expression of genes at the level of translation. And that can involve either these things called microRNAs, that's what miRNA stands for, microRNA, or siRNAs. And they both function to inhibit translation in some way. Okay, so both microRNAs and siRNAs function to inhibit translation, and they do it in a similar fashion. So they function to do that in a similar fashion. So what they're showing you here on the slide here is a microRNA. And what happens with this form of regulation is the microRNA or the siRNA has a sequence of nucleotides that's complementary to the sequence of nucleotides within an mRNA molecule. Okay, so because the sequence of nucleotides in this microRNA or siRNA is complementary, it's going to bind specifically to this mRNA molecule because it's complementary at this region. And then that binding will result in inhibiting translation in one way or the other. Okay. And just something you don't need to know for this course, but is useful, is that actually people now in labs will actually design siRNAs to inhibit gene expression of a gene that they're interested in. So let's say that you're interested in knowing what a gene inside a cell does. One thing to do is to see what happens to the cell if you prevent that gene from being expressed, because then what happens when the cell doesn't have that protein. And so what you would do is you would design a short RNA that has a sequence complementary to the mRNA of the gene that you're interested in, and then you insert that that RNA, inhibiting RNA, into the cell, and then translation is prevented, and then you can see what happens to the cell when you don't have that protein present. So this is a normal form of regulation in organisms, but it's also a form of regulation that we basically hijacked in order to study how genes work in an organism, because you can specifically design siRNAs to inhibit mRNAs that you're interested in. Okay, do you guys have any questions about the regulation by microRNAs or siRNAs? Okay, so again, we'll take another 10-minute break there, and then we're going to come back and do the lecture questions for this topic, and then move on to simple and complex patterns of inheritance.